Good evening, sir.
Good evening. Now we can start.
Yes, sir.
Good evening, sir.
Good evening. Screen is visible.
Yes, sir.
Yes, sir.
Okay. In our previous class, we have discussed about different genome analysis techniques such as DNA sequencing by next generation sequencing and Sanger sequencing, polymerase chain reaction, genome-wide associated studies, microarray, CRISPR-Cas9, and fluorescence in situ hybridization, and also brief about our proteome, different proteome analysis techniques, that is mass spectroscopy, two-dimensional gel electrophoresis, Western blot, protein microarray, X-ray crystallography, then isotope coded affinity tag. Then now we discuss about different sequencing techniques. First, we will discuss about the genome sequencing by clone method and whole genome by shotgun method. How this clone by clone means two term. One clone is there and another clone. That in the sequencing of this the genomic DNA, we have to do two different clones. How this is possible? We'll study. First, we isolate the genomic DNA from a cells or tissue. Then we fragmented by the different restriction endonuclease or DNase enzyme. Then this fragment should be in between 150 to 300 kilo base pair, because here we have to clone the this 150 to 300 kilo base pair DNA fragments in bacterial artificial chromosome. Bacterial artificial chromosome maximum clone is possible is 150 to 300 kilo base pair. So we have to fragmented the genomic DNA to 150 to 300 kilo base pair. Then this recombinant, then we insert this different DNA fragment to bacterial artificial chromosome, and then this recombinant bacterial artificial DNA then fragmented by the different um, DNase enzyme, which digests to give a product of 1.5 to 2 kilo base pair. Then this 1.5 to 2 kilo base pair, then we again clone in a plasmid vector. This plasmid vector is found in also in the bacteria. Bacteria have two DNA, two chromosome. That is, one is bacterial chromosome, another one is their plasmid, extra nuclear DNA. We call that is extra nuclear DNA. Then we clone that product in plasmid vector, and this recombinant plasmid vector then sequence, and we found the desired genome or gene sequences. If we go to the whole genome by shotgun sequencing method, then we isolate the genomic DNA from a tissue or cells, then ultrasonation by electric shock. Then the fragment is between 2 to 25 kilobase pair. Here is 150 to 300 kilobase pair. Here is 2 to 25 kilobase pair, and this cloning of DNA fragment into a plasmid vector. Here clone is once, and here clone is twice. So this is the method of clone by clone method, and this only whole genome by shotgun methods. And this recombinant plasmid contain our desired DNA is then sequenced using our Illumina sequencing platform. How these sequences happen by the after this recombinant plasmid. First, the chop product of this desired product of this DNA is aligned by uh, shotgun method, means overlapping sequence. How we found first restriction, we caught them in different restriction endonuclease enzymes, and this after the cutting by different restriction endonuclease enzyme, then we overlap the different DNA sequence. Suppose here is the DNA fragment 1, here is the DNA fragment 2, here is the DNA fragment 3, and here is the DNA fragment 4. Then we align them. You have to see here written was GCG. Then here starting is GCG. Then this alignment is possible. Then we have to call this is the first fragment. This is the second fragment. Then here the end is TTA, and here end start is the TTA. So we overlap these two sequence. Then we found the third sequence. Then after third sequence, here is the CCA, and here start is the CCA. If you overlap these two, then we have to found the next sequence. This is by overlapping with one another gene sequence to found a sequence DNA fragment by this clone by clone method or whole genome by shotgun method. What is the advantages of the clone by clone method? If you see here, we have to do the two times cloning method, which is mostly expensive, and here one time. This is the cheap method. uh, if we see the advantages, the clone by clone method of a genome sequencing is highly reliable as it follows prepared genome map for a assemble of fragments. And disadvantage is the clone by clone method of a genome sequencing involve the preparation of a genome map, which is costly and time consuming, because here we have to use two times and two vector. One is a bacterial artificial chromosome, and another one is plasmid vector. Here, so this clone by clone sequencing method is very costly and time taking, because due to the cloning process, it will take time. So this is the time taking process. And what is the advantage of whole genome by shotgun sequencing? The whole genome shotgun sequencing method, genome sequencing is cost effective and consumes less time, as it doesn't involve genome mapping and repeated cloning step. Here we have to clone only once, here twice. The what is the disadvantage? The whole genome shotgun method of a sequencing lacks accuracy, because when we align the first, second, third, and fourth, in this process we have missed any one of the fragments. So this is the this provides a disadvantage of lack of accuracy. Then next is how we know the open reading frame. First, what is open reading frame? In open reading frame, the DNA having one start codon and another one is stop codon, uh, which codes for a proteins, and that is called open reading frame. The in the open reading frame, as you know, the start codon is AUG, and the stop codon is TAA, TAG, TGA. Different types of three stop codon is there, and one is start codon. And in between the different nucleotides, it gives a protein by three nucleotides gives only one amino acid, and this is called open reading frame. How we have found? If we clone the sequence of the gene, as here we described, if we clone the sequence of the genes in a recombinant plasmid vector, then we sequence them. If we found the whole sequence, and we found, if we found any in this reading frame where we found only AUG, but this is the stop codon, then we found that this is our open reading frame having start site, because AUG is the start site of a transcription process, sorry, translation process. So if we found in the sequencing, if we found the TA or TAG or TGA, then this is the stop codon. One is the start codon, that is AUG, or another three is the stop codon, that is TAA or TAG or TGA. Then this is called a one open reading frame which codes for a proteins. Then, if we go to the mm, fluorescence in situ hybridization, that is FIS, then what is FIS? Fluorescence in situ hybridization detects and localizes specific DNA sequence using fluorescence labeled complementary DNA probe. Uh, for FIS, we have to know one gene previously, because if we do not know the gene sequence, then we cannot do the FIS, fluorescence in situ hybridization. How this occurs? Uh, for these probes one is the single stranded known DNA sequence or a fragment that complementary to the target DNA. If this is the target DNA and this is the probe, which is the known sequence that is complementary to the to this sequence, then the this probe, uh, if we consider this is a probe, then this probe should be labeled with a fluorescence dye. The labeling of probes can be performed either directly through the incorporation of fluorophore, or indirectly labeling with a hapten, which is conjugated with a fluorophore tag. The most widely used fluorophore in direct labeling include fluorescein isothiocyanate, rhodamine, AMCA, Texas Red. These are called fluorescent dye. While the most widely used hapten in indirect labeling, including biotin, digogenin, and dinitrophenol, these are called the indirect fluorescence dye. Then the target DNA. If we consider this is the fluorophore and this is the target DNA, fluorophore or a probe, this is the target DNA, then the target DNA is denatured using formaldehyde and heat. If we give the heat to the DNA sequence, and heat easily degrades the hydrogen bond between the A and T and G and C, then it will break to give rise to single stranded DNA. If the complementary, if the probe is complementary to this DNA, then this probe is bind to the DNA, and this probe is tagged with the fluorescence dye. After the binding, if we um, if we saw the DNA under the or UV light or normal fluorescence light, it will fluoresce color, and we confirm that this gene is this X gene is overexpressed or underexpressed in genome analysis. Then the target single-stranded DNA and probe are mixed together and incubated in hybridization buffer buffer, so as to allow the probe to hybridize with the sequence of target DNA that is complementary to it. After incubation, the unhybridized probes were washed away and hybridized probes are visualized under fluorescence microscope. We can only this fluorescent probe only bind to complementary to the DNA part or gene part, and if we see this picture under the fluorescence microscope, it will give rise a fluorescence light. And we found that this gene, if gene is overexpressed, and it gives more light, and if gene is underexpressed, it gives less light. So as over and underexpressed, we can found this gene is overexpressed or underexpressed. Several probes can also be simultaneously detected with different fluorochromes, showing the physical order on chromosome to be determined. And these fluorescence, the different fluorescence channels are called fluorochromes, including DAPI, that is blue in color, FITC, that is green in color, CY3, and rhodamine, these are red in color. These are different excitation range of visible light, that is blue, green, and red. If we go to the blue, then the blue is excited at 300 to 450. If we go to the green, then 450 to 520 nanometer. Then if you go to the red, then beyond 520 nanometer to 700 nanometer. For this range, we can detect by these fluorescence tags. Then if you go to the gene function using different database, because we have stored after the genome analysis or proteome analysis, then we have stored them in different genomic databases, and these databases give rise to what is the function of that protein or function of that genes. We can easily find out. If we go to the gene function databases, then these are essential tool for researchers to explore, unearth, and understand the role of genes in various organisms. These database contains information about gene sequences, expression patterns, how these genes are expressed, overexpressed or underexpressed, and their function, what kind of function of that genes, and interaction, how it interacts with different pathways, molecular pathways, and their association with disease. If a mutation occurs or insertion or deletion occurs, how these are associated with different types of diseases. And here are some key mm, gene function and databases. If we go to the gene ontology, this gene ontology database provides a structured vocabulary for annotating genes and their products with standardized terms related to biological process, cellular components, and molecular function. And these databases are freely available in our Google, and you can use this anytime. And if you input one gene there, it will give the details of the gene about their expression pattern, gene sequence, interaction, and what it leads to the different types of disease we can found. Then this databases is called gene ontology, and the key features is the hierarchical or organization of gene and function, and it is widely used for functional annotation of the enrichment analysis. If we go to the second one, that is UniProt, it is worldwide used databases where we store the different types of gene, millions of genes data inside this UniProt databases. This is a comprehensive protein sequence and functional information databases, that is UniProt, and the key features contains functional information about proteins, including the structure, function, post-translational modification, interaction, links to other resources like GOS, Assemble, and PTB database. For an example, if we go to the one database, my screen is visible.
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
Here I am giving one example, that is UniProt. This is one database. Here we can give one protein name, that is TP53. This is a tumor suppressor gene, that is TP53. TP53. You can here see the entry name of this different T53. I have given the query about TP53. This is a tumor suppressor gene, and it gives the all the data in human, mouse, bovine, rat, equus, rabbit, sheep, different species. If you choose only one human, this this is only given the humans, Homo sapiens species of TP53. How the one protein is 1009 times, means this protein is varied or mutated. Due to this mutation, different sequence will found in this gene. If I click to the TP53 binding protein 1, TP53 TG5, different TP53 mutated protein. If I click one, that is first one, then it will give rise to, it will check double strand break repair protein involved in response to DNA damage, telomere dynamics, all the function it will give rise, and it will also give rise the citation of this TP53 genes by different PubMed IDs. And here we also found the geo annotation, where it will found, then the cellular component, molecular function. Go to this, yeah. You can give where it is found, cytoplasm or kinetochore, where nuclear body, nucleoplasm. These are cellular function of this T53, and molecular function, it check only damaged DNA binding, histone DNA binding, any DNA binding it will check. Telomeric DNA binding, transcriptor regulator, and biological process may the DNA damage checkpoint signaling, because it check always the DNA, only DNA damage. If DNA damage will occur and it leads to mutation, and this causes cancer. All types of DNA damage, that is double strand break repair, double stranded break repair by a non-homologous enzyme. These are all the function of these T53 genes. Then if we go to the taxonomy, here we also found the different types of taxonomic analysis, and the gene name is T53 binding protein 1. And here that hierarchical organism, how this is uh, from eukaryotes to different metazoans, all the different animals have this T53 binding protein, and these are primary accession numbers. And if you go to the where it will found chromosome, which number of chromosome it will found, what is centromere position, kinetochore position of this gene, then we'll all the data we can found in this UniProt. And disease, what disease it will cause, a chromosomal aberration involving found in myeloproliferative disorder, cloning with eosinophiles. These different types of disease or data also you can find in this databases. And mutagenesis, different type of mutagenesis process also you can found here. And how the post-translational modification, different types of acetylation, methylation, different types of modification also found in that gene. You can input any protein in this UniProt databases. It will give rise all the data of that gene, where it will find how the pathway is maintained, which number of chromosome it will found, what disease it will cause, all the data. You can find all the data in this database. you can see how the different the length of this protein is 1972 and mass is molecular mass is 213574 Dalton, and it codes different proteins. These are called amino acid sequence of this protein, all amino acid sequence of the protein. You can find any of the protein in this database, and you can mapping from these databases. These are the large databases of uh, worldwide researchers are uses these databases, and also different what kind of disease it will cause. You can find anything, any expression pattern, where it will locate, and their taxonomical classification of that protein, and post-translational modification of that protein, and expression where it will express to nucleus, cytoplasm, or mitochondria, or Golgi body where it will express, and interaction, how this protein is interact with any other protein during this signaling, or hormonal or different kind of pathways, and the structure, what is the structure of this p53 genes and their family and domains, how the conserved domains present in that p53 genes, and the difference in the sequence of this t53, and the similarity protein of this t53 gene. We can found anything in this databases in UniProt protein resources. So this is content function, all the functional information about proteins, including structure, function, and post-translational modification and interaction. All we have seen in the databases, and these are links to other resources like Geo. First, we have seen the gene ontology, and different types of protein databases. Then next databases is called NCBI gene databases. This is a protein databases, and here this one is the gene databases. In this NCBI gene database, that provides detailed information about genes and their function, pathways, expression, and literature references. As we know, everything we will found in this NCBI gene databases. If we go to the NCBI gene databases, NCBI, this is the largest national center for biotechnology information. This is the leading NIH databases. It is totally developed and utilized by United States National Library of Medicine. Every in worldwide, all the researcher used this NCBI databases for all gene protein data analysis. If we go to the gene, then here we found same TP53 gene. You can check what is the function of this T53 gene. Okay, this this is the TP53, that is tumor protein 53 gene. And if we go to the here, it will gives rise where the location that is located in chromosome number 17, because this is the genome or gene or DNA. This will give rise to all the chromosome references where it will found in Homo sapiens, that is 17, in ratus that is 10, in zebrafish that is chromosome 5. Everything you will find here, and the sequence of the genome, we can also find the all the sequence of this T53 gene here. Okay. Yeah. You can find here the summary of this T53 gene, that is Homo sapiens T53 genes, and it codes a protein, that is tumor protein P53. That is a 53 molecular weight protein, and this gene code encodes a tumor suppressor protein containing transcriptional activation, DNA binding, because it binds to DNA to check whether DNA is damaged or not. The encoded protein responds to diverse cellular stresses to regulate expression of target genes, thereby inducing cell cycle arrest, apoptosis, senescence. All the function you can find every gene. You can input any gene and it will give rise what is the function of that particular genes. All the function you can find also here, and what is the location of that gene, that is in Homo sapiens, that is chromosome number 17, and here this is called chromosome number 17. What is the open reading frame, how it starts and how it ends, all the data you can find in this NCBI databases. And these are the references where the people referring about the different T53 abnormal expression, what it will cause, dysregulation in P53 in breast cancer, positive factors and treatment. These are all references citing for this protein P53 functions. These are called large databases. One is for protein is UniProt and another one is NCBI gene. Then third is Ensemble. This a genome browser that provides detailed information about same gene function, expression, regulation across multiple species. And this key features is gene annotation, comparative genomics, and variation of data, visualize gene structure, splicing patterns, how this splicing will occur. This is a single splicing or multiple splicing mechanism, and regulatory regions. The then next one is Kyoto Encyclopedia of Genes and Genomes. This is only for the biological pathway, how these proteins are involved in different pathways. This is a key database that integrates gene function with biological pathways and molecular interaction and disease information. These cake pathways maps that link genes to metabolic and different signaling pathways, tools for pathway enrichment analysis and gene set analysis. This is for pathway enrichment and gene set analysis. And sixth one is OMIM, that is online Mendelian inheritance in man. This is important for human genes and genetic disorders or linking genes function with diseases. You can use any databases for your you can take any examples of genes or protein, you can put them in these different types of databases and it will give all the uh, data like signaling pathway, protein interaction, genetic disorders, or linking genes and function. If you go to the seventh one, that is source tool for retrieval and interacting genes or protein, that is string. In this database, you will found this KEGG and also the reactome. This KEGG and reactome is only for biological pathway. If you know which pathway this gene is involved and causing different types of disease, we can target that pathway to uh, kill different types of disease. So we have to know which pathway this gene is involved. For this, if we go to the one example of this string database, here we can put on one query. Here some proteins we have taken, different types of proteins, how they are interact. We can put here different types of here I have taken 3, 4, 5, 6, 7 proteins, and here organism is Homo sapiens. Select, then search. How these proteins will interact, we can find in this string databases. These are the description of these different 6 7. I have inputted the 7 6 7 different types of proteins, and then I'll go for continue. See how these genes are interact with each other. It will give by different coding of the different coding of this line, and how we can describe here. Suppose here you can find the how these genes are responsible and interacted, and these genes are interacted to form cellular response to indole 3 methanol signaling, and these are response to DNA damage response, signaling transduction by p53 class. These are related to p53 signaling. Then histone STO monoubiquitination process and circadian behavior, and molecular function is protease binding, and cellular component is lateral elements. If we go to the how here we have described about reactome and cake. These are giving the how their pathway map that link genes to metabolic and signaling pathway. Here also you can see the visualized pathway related to human biology. You can find here the cake pathway. If you mutated or overexpressed or underexpressed these 7 genes, it will cause thyroid cancer, and it will dysregulated the homologous recombination. And also these are, if these are the major genes, this BRCA1, TP53, PTN, CDH1, BRCA2, and STK11. If you mutated this gene, it will cause any cancer like thyroid cancer, endometrial cancer, bladder cancer, or melanoma cancer. If you click this converted to a red color, means in thyroid cancer which genes are interacted? Only two. One is T53 and this CDH1. If you mutated these two genes, then the it will cause a thyroid cancer. If we go to the endometrial cancer, which one is involved in endometrial cancer? I'll deselect this, and yes, in endometrial cancer you can find the in mutation of T53 gene, PTN, or CDH1. If you mutated these 3 genes, and it will cause endometrial cancer. We go to the next bladder cancer. Then in bladder cancer also CDH1 and T53 genes. In melanoma, skin cancer, if you go to the skin cancer, then also skin cancer. Here you can find T53, PTN, and CT. TP53. This is the major gene which is responsible for all type of cancer. If this gene is mutated or overexpressed or underexpressed, it will cause any type of cancer. Here in string databases, you can find different pathways. In that pathways, which proteins are involved. In reactome pathways, also you have found different different pathways. How this homologous recombination is dysregulated, you can find in this string databases. Then the flybase is for a database for only Drosophila melanogaster gene function, genome data, and expression pattern, because we have used different model system like Drosophila melanogaster, or rat, or mouse, pig. These are called different models. These, because we have used these different model system, because they have similarity, 95% similarity with our genome. So we have studied different model organism like Drosophila melanogaster, mouse, rat. In this flybase, you can find the focused on Drosophila gene function and development, and cross referenced with other genetic functional different functional databases. Then the last one is Human Protein Atlas, a database that provides information about protein expression and localization in human tissues, linking gene function to their protein products. And, the website is here, Human Protein Atlas. You can click any of this website. After I will give the PPT, you can use this website for know the gene or protein function of any. You can input any gene or any protein to study their function or their any interrelation between the gene signaling or any pathway study or any function of that gene. Then these are the gene function databases, and next one is the databases in protein sequences and protein structure determination, because these databases only known for the different protein structure. As we know, in protein have primary structure, secondary structure, tertiary and quaternary structures. And the structure and sequence of a protein can be determined using various types of databases. A database is organized collection of data that is set up for easy access, management and updating information, because all the information you can find in these databases. The protein data bank, that is PDB, is the main source for 3D structural information on proteins and the biological macromolecules such as DNA and RNA polysaccharide. Because as we in our book we have see the only two-dimensional structure of the protein. If we go to the secondary structure is the combination of alpha helix and beta pleated seal. How this structure you will find the in this PDB databases, you can find the three-dimensional structure of that uh, any proteins you can find the 3D structure. Then for structural data analysis, it is easy to obtain the information from one of the protein structure databases rather than collecting the data from PDB. They provide detailed information on specific structural motifs, because these motifs and domains are called super secondary structures, and these super secondary structure of this protein you can find also in this PDB databases. Um, and these are various examples of protein databases, include MODbase, GNA library, PDB Lite, PDB Sum, Oracle Certified Associated, that is OCA databases, orientation of proteins in membrane databases, that is OPM, PDBTM. These are different types of databases for you can find the protein three-dimensional structure of the protein. If we go to the one example of protein data bank, that is PDB. This is the PDB homepage. Yeah, you can search. Huh. In PDB data bank, that is protein data bank. In the PDB data bank, you can find the structure similarity search. You can similarity check of different types of genes. Then chemical sketch tool, you can draw the structure of the different proteins. If you new entry, if you found a new protein, then you add that protein into these databases, and you can say sequence similar search. The sequence similar of different proteins, you can align different proteins with each other, and you can find the sequence similarity between these two proteins. And different browser annotation, chemical similarity search, you can use any of these databases, and you can visualize the 3D visualization of the PDB structures. This Yes, here you can see the three-dimensional structure of this uh, uh, T53 protein. Here, these are called, this arrow you have seen in the different books. These are called beta sheets, and these spiral are called alpha helix. These are alpha helix. These are connected with beta pleated with a torn, and these are called secondary structures of the proteins. You can, these are the crystal structure of the P53 protein, and here all the related documents or reference you will find in this protein data bank. You can uh, you can use any type of protein and put it here the 3D structure, and it will give the three-dimensional structure of the protein. Then we go for the basic local alignment source tool, that is BLAST. This BLAST is you based on the type of sequence to be aligned, because we have to check the alignment between the two different genes or different RNA or DNA or protein. You can check anything using this BLAST tool. This is BLAST P. This compares by amino acid query sequence against a protein sequence databases, that is BLAST N is compare for nucleotide. This is comparison of two different proteins having what type of amino acid comparison between the two different of protein. You can check the two different of genes or nucleotide sequence by this BLAST N, and then TB LASTN that compares protein query sequence against a nucleotide sequence. If you put the protein or amino acid sequence, it will give rise the nucleotide sequence using the this TB-LASTM, and that is BLASTX. This is compares nucleotide query sequence translated in all reading frames against, but here you can put the nucleotide sequence and it will give rise the proteins. And these are the basic local alignment source tool, and these programs are used for a database for nucleotide, this BLASTN for nucleotide databases, and query also you can give the nucleotide sequence. And here the in BLAST P, you can, the database is only for protein and your query also be protein. And in TB BLASTN, and this is for translated nucleotide, you can put here nucleotide and it will give, you can, these databases is for only nucleotides and query. You will put the protein name and it will give rise the their nucleotide sequence. Similarly in BLASTX, if you input the translated nucleotide sequence and it will give rise the protein. Then these are the based basic local alignment source tool to study the protein structure or DNA structure and function analysis and determination. If we go to the BLAST tool, yes, here you can nucleotide BLAST. This is BLAST X. This is TB BLAST, TB BLAST N. This is protein BLAST. You can choose anything. You can click anything, then it will go to the alignment, and you will put your sequence or nucleotide sequence. It will give rise to a protein sequence. If you go to the BLAST P, then you can put here your protein sequence. This BLAST X, this TB BLAST N, this is the TB BLAST X. Here also you can use these. These are the basic local alignment source tool for this database in protein sequence or DNA sequence analysis. Then after these are called bioinformatics tools. These are all are bioinformatics tools, and all the data are stored in these all the different types of databases. But if we go to the uh, disease analysis or gene expression analysis, you can use different techniques for gene expression profiling. That is one is for genomics or gene expression profiling, you can study the development, differentiation, how disease progresses, how pharmacogenomics, disease management, how we'll manage that disease, then disease classification, then molecular staging, nutrigenomics, treatment prediction, diagnosis, new target identification, personalized medicine. Simple example is during the COVID phase, we have analyzed the SARS-CoV virus using quantitative real-time PCR methods. I will, according to their viral load, we can go for the diagnosis or treatment prediction. How will treat that patients, and the patient will be cured or not. You can all these techniques for gene expression profiling. Any type of gene expression profiling, we can use this quantitative real-time PCR. This is quantitative method. Quantitative means how much amount of the gene is present, and this gene is basal level expression or overexpressed, or that is underexpressed. We can find out by this quantitative real-time PCR method. How this PCR will work? This to perform a quantitative real-time PCR or qPCR, the samples are collected from different sources in which we want to study the expression pattern of the any transcript. Means transcript means that is mRNA. And this qPCR machine has a fluorescence detector in addition to the regular components of a PCR machine. And this qPCR uses the different types of primers designed from the target genes in a PCR reaction to analyze the transcript levels. And the fluorescence detector measures the fluorescence at every cycle, and the transcript level is calculated by considering the variation in intensity observed after each cycle during the PCR. How will do the PCR? The steps of the PCR is first we have isolated the double-stranded DNA from a cell or tissue. Then we denature them the double-stranded DNA using heat at 95 to 95 degrees Celsius. Then we have add one primer, because the primer and DNA polymerase is required for this annealing or PCR reactions. And we add one primer, and then DNA polymerase anneal these sequences of this 5 prime to 3 prime direction, or 3 prime to 5 prime direction. This is called annealing stage. For anneal, because if you do not do the annealing step, then the primer will not bind to the uh, one strand of the DNA or gene, or other strand of the DNA or gene, because we have to increase the copy number. Because if you know the gene is very low amount expressed, then we have to do the quantitative real-time PCR to increase the copy numbers of that DNA or particular genes by this quantitative real-time PCR. Then extending stage, in extending stage, DNA polymerase is used to uh, extend the primer to 5 prime or 3 prime directions. And this is the how we quantitate. And during the quantification process, the double stranded DNA template is denatured, and then denatured into two different strands, that is 3 prime to 5 prime and 5 prime to 3 prime direction. Then we add a primer by annealing, and then this primer is annealing by the DNA polymerase for extension. After the extension, we use a fluorescence label tag, that is cyber green, and this cyber green binds to the minor group and major group of the DNA, and it give a fluorescence. And after this sequence, after here we have started with one template. Here we have found two different templates by primer extension by DNA polymerase, and in this template 1 and DNA template 2, after the major and minor group will formed in that DNA, this fluorescence dye will bind to the minor and major groups, and it will gives a fluorescence, and that fluorescence amount is captured by the PCR machine, and to know this is a one strand or it is a two strand. If it is a two strand, and it will give more fluorescence, and this fluorescence level is um, captured in a real time, and it will give the quantitative how much the RNA is converted to DNA, or how much DNA is copied to different numbers, and it will increase by 2 to the power n. If we go to the 2 cycle, then it will 2 DNA template will give rise to 4. If it goes to 4 cycle, and it will give rise to 16 number of different DNA fragment, and these 16 number of different DNA fragment will give a some fluorescence amount, and this fluorescence amount dictates how much the techniques most widely used quantitative real-time PCR PCR method, and it is used for different disease diagnosis or any other gene expression analysis. Then next one is nuclear run-on assay. What is nuclear run-on assay? A nuclear run-on assay is a method for measuring rate of transcription of specific genes in the nucleus of a cell. It is only targeted to the nuclear region expression, and it is considered the gold standard for measuring the transcriptional activity of a promoter. And for this is only targeted for promoter region, because promoter is required for the start of the transcription process. What is transcription? Now, this is the copy of a DNA. Sorry, DNA is converted to mRNA or messenger RNA or mature RNA. This is called a transcription process, and for this transcription process, a promoter is required for start of the transcription. How will know the promoter is present or absent? We can use this nuclear run-on assay. How this will happen? We can take a normal cell or a cancerous cell, and then this cell is cultured in a culture media, and we harvested or isolated the mRNA from this normal cell or cancerous cell, and then this mRNA, by using reverse transcriptase enzyme, we converted to the cDNA, and this is the cDNA, and we can tag with a different fluorescence, that is red fluorescence probe or a green fluorescence probe, and we can hybridize the cDNA probes to oligo sequence on a micro array, and this is run by a micro array method, micro array techniques. And here in black color, we know the known sequence of this gene, and if it red color will bind to a particular this known genes in the microarray tag, we will know that this gene is bind to a particular sequence in the microarray, and it will give rise to the overexpression or underexpression pattern of that particular genes by the hybridization techniques. And this microarray uses chip which contain oligonucleotide corresponding to the transcript of a cell, and this comparison between two cell types differing in their properties and can be performed by extracting the RNA and converting it to the cDNA with two different fluorescence tags, followed by mixing and hybridization. And this color pattern on the plates is used to find out the differentially expressed transcript in the cell types. What is differentially expressed? Means the differentially expressed means that gene is overexpressed or underexpressed. Always in our cell, all the gene is expressed in a basal level, And if it goes beyond the basal level, above the basal level, that is overexpressed, or if we go the expression is below the basal level, that is underexpressed process, and this is called differentially expressed transcript. Similarly, we can use also northern blotting for the expression of a specific or a particular gene. We can use by using this northern blotting technique when the expression of specific genes needs to be analyzed and the sequence of the gene is known. It is possible to use the northern blotting for expression analysis. It is more useful in those conditions where the cells are transfected with a gene or a protein gene for protein expression. How we'll do in samples, different types of you can use different cells or normal cells or cancerous cell or different type of tissue. Then we extracted the RNA and the electrophoresis. In the electrophoresis process, we use the agarose gel, and in this agarose gel, we separated this RNA according to the charge to mass to charge ratio. uh, in upper of the this gel we can found large molecular weight, and in lower of the gel we can found the small molecular weight, because smaller the molecular weight it moves faster, and the larger the molecular weight it will move slower because due to the retardation process. Then this RNA is separated by size with mass to charge ratio. Then it will transfer to the nitrocellulose membrane, and this RNA fixed to the membrane by UV and heat. And if you go give rise the UV and heat, the membrane will hybridize with the the this membrane then hybridize with different label known probes, and this RNA will bind to the label probes, and these label probes will visualize in RNA on a X-ray film. This X-ray film will in X-ray film like X-ray. If you go to the if you go to do the X-rays process, how it will find? Now these will penetrate the X-ray is penetrate through this X-ray film, and where the probes will found, it will give a black sign, and this black sign will found the expression of that genes. It will not hybridize the proper gene with this RNA, then it will not find in this X-ray film, and this is called the northern blotting processes. Then the next one is RNA sequencing method. How we'll do the RNA-seq, and this if you go to the RNA sequencing method, this is a technique that use next generation sequencing to analyze RNA molecules in a biological sample, any biological sample that is tissue or cell. It's a powerful tool for studying gene expression or transcriptome. Transcriptomes mean any RNA, that is long non-coding RNA or rRNA or small nuclear RNA or mRNA. These are called transcriptomes, whole RNA, and used in wide range of application including cancer research and disease diagnosis. You can use this sequencing method any cancer research or disease diagnosis or SNP, single nucleotide polymorphism detection. Then what we'll do in the RNA sequencing, isolated cell or tissue population, you can extract the total RNA, and in the total RNA you will found all the transcript, that is long non-coding RNA, rRNA, small non-coding RNA, mRNA. And in this extraction, higher abundance of the ribosomal RNA we will found, then it will hinder the small non-coding RNA parts, so you have to degrade this ribosomal RNA, and this mRNA having a poly A tail. You can select this poly A tail by using oligo DT primer. If you add the oligo DT primer, that is TTTT, it will bind to this mRNA and it will synthesize to a cDNA product. If we ribodepleted this product, then you will find only the different small nuclear non-nuclear RNA, and you can also choose the size selection by using the different chromatography method. You can use different size selection, and after the isolation of these all the transcriptomes, you can convert it to cDNA using reverse transcriptase enzyme, and it will convert it to cDNA, and this cDNA you can use for a sequencing or a in Illumina Sec platform by overlapping with these uh, different types of genes. You can construct a sequencing library, and this PCR amplification and sequencing occurs in a Illumina Sec platform. This is called RNA sequencing or RNA-seq method. Then if we go to the different techniques, these are expression of genes or their product, that is called RNA, because from DNA we can found the RNA, and how much RNA content it will give rise to the proteins. Then these are the identification or analysis of genome or transcriptome, and here we can analyze the techniques for proteome analysis. In proteome analysis process, first uh, we have studied the Edman degradation process. These are called N-terminal determination of a protein, because protein have one is N-terminal, another one is C-terminal. If you go to the N-terminal determination, in Edman degradation process, Edman uses one chemical, that is phenyl isothiocyanate, is the primary reagent used in Edman degradation process. And in Edman degradation process, here is the amino acid having N terminal NH3 plus, and here is the C double bond O, and here is the R, and here is the N terminal attached with another amino acid or peptides. And this, after using the phenyl isothiocyanate, it will bind to the N terminal of the proteins or peptides, and this is called N terminal detection of proteome. Then it will give rise to a one is phenyl isothiocyanate derivatives, and this phenyl isothiocyanate derivatives is cleaved by using the hydrochloric acid, because this is the strong acid. It will break all the peptide bonds, and it will break the peptide, that is NH3N and peptides, and this is the thiazonolone, and the remainder of the peptides, then it will give to the another sequenter machine is there. It automatically go for the next amino acid detection, and this thiazonolone derivatives is unstable, and this unstable product give rise to a phenyl thiohydrantoin derivatives, and these phenyl thiohydrantoin derivatives using the uh, using the chromatography methods, you will find what is the amino acids by chromatogram, and this is the N terminal determination only in the using the PTC adduct, and this PTC only bind to the N terminal of the amino acid residues, and this is the Edman degradation process. If we go to the mass spectroscopy determination, in mass spectroscopy determination, we can choose the mass of the peptides or proteins. In this mass spectroscopy determination, here is the samples. This sample contains different sized mass amino acid residues or peptide or protein residues, and this laser disrupt to form a charged particle, and this charged particle is passed through an analyzer, that is called top time of flight, the time of flight, that is top. In this top, this is the in this top, this is the movement of this peptide is through the in an electrical field, and the slower, the smaller the peptide length, it will move faster, and larger the peptide length, it will move slower. And the smaller or far larger particle detect by a detector in the mass spectroscopy, and it will in the mass spectroscopy, this detector, after the detection, this peptide will be blast in the uh, uh, databases, and it will give rise to the which type of protein or nucleotide. And in this multi-top mass spectrometry, the protein sample embedded in an appropriate matrix, but this is ionized by application of this laser beam, and thereafter the electrical field accelerate the ions from through the flight tube towards the detector. The lightest ion, that is the smaller the peptides, it will move faster and fast detector, and simultaneously the time of flight is measured by the clock triggered by ionizing laser pulse. And smaller the peptide length, it will move faster, and the detector will detect. And after the detection of the smaller peptide fragment, it will be aligned in the databases using blast p software. And this blast p software will tell this is the which fragment of that protein. It will give rise the all alignment protein analysis, and it will also give the whole protein analysis. Then after the mass spectrometry process, then we go for the SDS page analysis, that is sodium dodosyl sulfate polyacrylamide gel electrophoresis. It is a electrophoretic mobility process, that is in the presence of the electrophoresis, the protein will move faster in an electric field. We can separate the uh, protein by their molecular weights or molecular shapes. If we go to the if we extracted the whole protein in an sample from a tissue or a cell samples, and in the cell or protein sample, the protein having in its tertiary or quaternary stage, then we have to lyse them using different type of beta, like beta mercaptan ethanol. Beta mercaptan ethanol breaks the disulfide bond of the peptide, and this disulfide after the breakage of the quaternary structure disulfide bonds, all the proteins are converted to primary structure. After the conversion of the primary structure, this sodium dodosyl sulfate, this is a anionic dye, and this will bind to the protein by 2 by 2 amino acid. Every 2 amino acid, one SDS will bind, and this will give rise to a negative charge to that protein. Here you have the heated the protein, because heating of the protein will break the non-covalent bonds in proteins. And for covalent breaking, we use beta mercaptan ethanol, and it will give the all the proteins into a negatively charged. Every 2 amino acid, one SDS is bind by hydrophobic interaction or bind to the arginine residues of the sulfate groups, because this have a one sulfate group that is minus, and it will bind to the arginine, that is a positively charged amino acid of that protein, or a sodium dodosyl, that is a alkyl 12 carbon alkyl group, sodium dodosyl having a 12 carbon alkyl group, and it will bind to the hydrophobic amino acid of that protein, and it will bind every 2 amino acid one SDS, and it will give rise to one negative, all negative charge to the protein. Because of the negative charge, it will move faster in a stacking and resolving gel. In this stacking gel, the pH is 6.8, and in resolving gel pH is 8.8. And in the running buffer, we can use chloride ion, glycine ions, and the proteins. And chloride glycine ions move the protein towards the uh, positive charge, because this protein have negative charge, and it will move to the positive charge. And after the electrophoretic mobility, then these are called the different samples of protein loading. The first lane is the molecular weight of the known molecular weight of the protein, and this known molecular weight of the protein give rise to the unknown molecular weight of the protein molecules. And if you load any protein inside this gel, it will give a band, and this black band give rise to, suppose this is 220 kilodalton, this is 100 kilodalton, this is 150, no, 80 kilodalton, 50 kilodalton, 30 kilodalton, 20 kilodalton, this is 10 kilodalton. You can find the unknown molecular weight of the protein. If you load any sample, and it will give a band, and you can tell them that this is called 50 molecular weight band, or this is 80 molecular weight band, or this is 120 molecular weight. You can know all the molecular weight by this SDS page methods. Then if you go to the Western blotting, you can in Western blotting you can see the expression of any type of protein. In QRT PCR, you know the expression of the all RNA molecules. Here in you can see the expression of any type of proteins. If you go to the Western blotting workflow, then similarly take the cellular protein of different condition, unfolded or coated with negative charge with SDS. You can give the negative charge to the protein. Then you in you can do the gel electrophoresis by loading this protein into the mm, this polyacrylamide gel. These are inert gel, and these proteins are separated. This is the known molecular weight protein. How see how these proteins are separated according to the molecular weight. The smaller proteins are move faster, and the larger proteins are here. If we choose, this is called 200, this is 150, this is 100, this is 80, this is the 30, 20, 10. If this is 200, and you can find the molecular weight of the above these three are called 200 molecular weight. This is 200 kilodalton protein. If you go to this is 150 kilodalton protein. If you see this is 100 kilodalton protein. This is 80, there is no protein in 80 kilodalton. This is 50, there is no protein in 50 kilodalton. uh, you can find in 30 or 50. In between this is maybe 40 kilodalton protein. These are 10 kilodalton protein. You can find out the molecular weight of the also in Western blotting process. Then you then you can transfer this protein to a PPT membrane, because in Western blotting this is a immunofluorescence techniques, and this is you can bind. If you go to the check whether my TP53, this is tumor suppressor protein, TP53 is overexpressed or underexpressed in cancer condition. If you go to find out this, then you go for after the separation of all the proteins, then you transfer that protein to a membrane. That is, the membrane is positive charge, because all the proteins are here in negative charge because binding of this SDS molecules. Then this in the membrane you can add uh, a primary antibody against the TP53 genes, and this primary antibody bind only to the P53 genes. And against this primary antibody, then you can add the secondary antibody, which binds to the FC region of this primary antibody. And this secondary antibody contains a fluorescence tag, and this fluorescence tag give rise a signal, and this signal you can see like this is overexpressed, this is mediumly expressed, this is underexpressed. You can compare with the normal cell as well as cancerous cell or other cells. You can find the expression of any proteins inside the cells by this Western blotting workflow. These are all the workflow of the gene as well as protein analysis. Uh, we can brush up every techniques. First, we have studied the genome sequencing by clone methods. Here we have taught about the two different types of cloning methods. Here only one time of clone. This is the sequencing of genome. These are two different methods. One is clone by clone, and another one is gene genome by different shotgun techniques. Here also we know the open reading frame, that is the protein coding region, that is a protein coding region of a DNA that having a one is start codon, another one is stop codon that identify the protein coding sequences. Next one is fluorescence in situ hybridization. By in fluorescence in situ hybridization, we detect and localizes specific sequence using fluorescence level complementary different DNA probes. And here we have discussed about the all the gene function using different gene ontology or different databases where the data, all the data are stored of the known gene or proteins like gene ontology, UniProt, NCBI gene, ensemble, cake pathway for pathway analysis, OMIM for catalog of human genes or different types of genetic disorders. Here also in string we can analyze the pathway. Here reactome we also analyze the different pathway fly basis for only Drosophila melanogaster gene functions. Then here we described about human protein, different protein atlas, and also uh, here the different databases for protein sequence and protein structure determination. And here the basic local alignment source tool, that is BLAST, different BLAST, that is BLAST N for nucleotide databases, and query term only nucleotide. This is the BLAST P, that is databases only for protein. And here also you can give the protein query. Here the TB blast in, here the database is only for nucleotide, but you can put your query in protein and it will give rise to the nucleotide bases. And here is the BLASTX. Here the databases is only for protein, and you can input your input is your nucleotide, and if you input the nucleotide, it will give rise to what kind of protein that is. Then we have discussed about different techniques for gene expression profiling. The most widely used technique, that is quantitative real-time PCR, and this is the fluorescence dye, that is cyber green we use for this, no, for um, the quantitative RNA expression analysis using this PCR methods. And here also described the different nuclear run-on assay and northern blotting for gene expression analysis. And here is the RNA sequencing. Nowadays it is widely used to know which type of RNA is required for which type of function in our cells or genes. To know this, we have to do the all whole RNA sequencing. This is the RNA sequencing by different, we have extracted all the RNA and then it converted to cDNA. Then we can do the PCR amplification, and using different Illumina sequencing platform, it will give rise all the sequencing processes. Then we have described about techniques for proteome analysis, how we can protein analyze. If we analyze a peptide, then we can use Edman degradation process. In Edman degradation process, this is for N-terminal determination. It can give rise to the N-terminal amino acid each time. A fast detection of N-terminal analysis. Then this peptide you can add then one PTC, phenyl isothiocyanate, then the second amino acid it will detected by this process. And then third it will detected the third amino acid by this 1, 2, 3, 4 detection. You can find out the all the sequence of the amino acid. In mass spectroscopy spectrometry process, you can use multi-tau mass spectrometry. This is the embedded in a particular matrix. The matrix is inert, and then you can use laser for disruption of all the peptides, and it will travel through an electrical field which accelerate the ions formed through the flight tube and towards the detector, and the highest ion arrives first because the charge to mass ratio, this is the smaller ion will move faster and larger ion moves slower, and the detector then detect the peptide, and then it will align with the blast fish softwares, and it will give rise to the which protein or which peptide you can will find out this mass spectrometry processes. And also you can use for the to know the molecular weight of the peptide, you can use sodium rhodosyl sulfate polyacrylamide gel electrophoresis. In this process you can give a negative charge to the peptide or protein using this sodium rhodosyl sulfate, and this negative charge protein will separated according to their charge to mass ratio in an electrical field, and this is the gel peak of a polyacrylamide gel electrophoresis. Here is the known molecular weight of the protein or peptide, and you have add here in every oil you can add these are called oil, different oils. You can put your protein samples here, and it will separate it according to the molecular weight, and it will give rise to the a band. This band is called the particular molecular weight of the protein or a peptide. In western blot technique you can know the expression pattern of the protein or enzymes. You can use different antibody immunofluorescence dye, that is primary antibody and secondary antibody, and this primary antibody is bind to your desired protein, and after the primary antibody you can put the secondary antibody, and this secondary antibody will bind to the Fc region of the primary antibody. After the secondary antibody bind to the primary antibody, it will give a fluorescence, and this fluorescence will you will see this fluorescence in a uh, chemiluminescence dog or chemidog. In this it will give the medium, low or high fluorescence, and this fluorescence result you will show this the protein is overexpressed or underexpressed with respect to any experimental condition. These are the expression profiling of all the genome as well as DNA, RNA and proteins. Doubt कार अछि?
तो फास्ट स्लाइड रे सर जो कहले कि आपण जो ओवरलैपिंग होची सर से ओवरलैपिंग रो सर सिग्निफिकेंस ता कोन?
ओवरलैपिंग ର सिग्निफिकेंस ହେଲା ଆମେ ଯେତେବେଳେ କଣ କରୁନା ଯେତେବେଳେ ରିକମ୍ବିନାଣ୍ଟ ପ୍ଲାଜମିଡ୍ ବନଉ ଏଇ ପ୍ଲାଜମିଡ୍ ରେ ଯେତେବେଳେ ଆମେ ଗୋଟେ ଜିନ୍ କୁ ରଖିଛୁ ଏଇ ଜିନ୍ କୁ କଣ କରୁନା पर्टिकुलर रेस्ट्रिक्शन एंडोन्यूक्लियर କାରଣ ଆମେ ଏଠି ମିଲିୟନ ଅଫ କପି ପାଇବା डिफरेंट डीएनए फ्रैगमेंट्स ର बट आमे केमती जानिबा जे एई जो डीएनए फ्रैगमेंट्स अछि आमे ताको अलाइन करिबा केमती अलाइन करिबा तो सेतिपई आमे कोन करूना रेस्ट्रिक्शन डिफरेंट रेस्ट्रिक्शन एंडोन्यूक्लियस यूज करिकी ए डीएनए जो हमर ए जो रिकॉम्बिनेंट प्लाज्मिड को फ्रैक्शन कर दो फ्रैक्शन करले कोन हबो ना फ्रैगमेंट करले तापर आमे जानिबु केमती कोटार फर्स्ट फ्रैगमेंट कार सेकंड फ्रैगमेंट तो सेतिपई जदि आमे टू डिफरेंट डीएनए एंडोन्यूक्लियस यूज करथबु सपोज सीएटी जी परटारु काटिची आ गोटे से नेक्स्ट एंडोन्यूक्लियस काटी नथिबो आ जी पर से काटथिबो टी पररु काटथिबो जे जी पररु काटिबो ताले से कोन करिवो ना रेस्ट्रिक्शन एंडोन्यूक्लियस सी गोटे नोन पर्टिकुलर सीक्वेंस रु काटिबो जो सीक्वेंस ता जेतले तमे सीक्वेंसिंग करिवो तमे जानिबो सपोज जदि एजीसीजी थिबो ताले गोट ए रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम ही एठी ओनली काटिबो फास्ट सेकेंड आउ गोटे यूज करथबो जेमति कि से आउ गोटे जगह रे नोन सीक्वेंस रे काटथिबो तो हेले एमती हमको बहुत सारा फ्रैगमेंट मिलिबो जो फ्रैगमेंट को हमें कोन करू ना अलाइनमेंट करू ओवरलैप करू गोटे ऊपर गोटे को ओवरलैप करू तो कले कोन हबो खाली जीसीजी नो मे बी एटी एठू भी स्टार्ट हेइथै पारे ए टी सी सी ए जी सी जी तो हेले कोन हबो ना आ गोटे फ्रैगमेंट एठी ओवरलैप हबो पुनी जदी ए नेक्स्ट फ्रैगमेंट ता मे बी एटी टीटीए रु एटी नोन सीक्वेंस बेली टीटीए रु हमें लिखि छु नले मे बी एटी टीटीए एठी भी अछि एठू भी अलाइनमेंट हे स्टार्ट हेइ पारे तो नेक्स्ट वन इज सीसीए जदी एठू मे बी सीसीए थिबो जदी एठी सीसीए थिबो तले एठू भी अलाइनमेंट हेइ पारे तो अलाइनमेंट हेले कोन हबो बेनिफिट्स ना तमे गोटे पूरा नोन कॉपी ऑफ डीएनए फ्रैगमेंट्स पाई परियो जो तरे कि तमरो जो डिजायर्ड थिबो जीनोम टा सेटा तमे जानि परियो एटा रे कोन ना होल जीनोम सीक्वेंसिंग करंती माने टोटल जीनोम को तमे सीक्वेंस करूछ सेटा रे ए मेथड यूज करा जाय
सर जो डीएनए टेस्टिंग करा जाए जो माने गोटे पिल्लार तार पेरेंट्स सहित मिसेया पई आ भितरु माने किछि प्रोसेस माने इन्वॉल्व अछि
सेटारे डीएनए फिंगर प्रिंटिंग करा जाए सेठी सबु माइक्रो सैटेलाइट और मिनी सैटेलाइट देखंती जोटा कि हमें काली पढुतिले कि रिपीटेड सीक्वेंस थिबो जोटा कि पेरेंट रो ऑफस्प्रिंग आस्थिबो कि नले जदी कार से जदी थिबो जदी कोठी के रेपिस थिबो तार जदी हेयर थिबो कि फॉलिकल थिबो नेल थिबो कि ब्लड स्मीयर थिबो कि सीमेन स्मीयर थिबो तो तमे ताको कोन करि परिबो ना तमे ताको एनालिसिस करि परिबो पैटर्न जानिया पे तमर सपोज तम जीनोम को मु जदी गोटे रन कली जेमति हमें एठी देखुतिले सपोज मु एठी देखौची सपोज तम जीनोम को मु रन करिछि सपोज गोटे एमती पैटर्न आसीछि ए जो पैटर्न देखुचो एटा गोटे पैटर्न ऑफ तम जीनोम रो आसीछि फ्रैगमेंट हे तम जेतबेले मु नेक्स्ट तमरो जेतबेले मु पेरेंटिंग टेस्टिंग रे सेतबेले जेतबेले आ गोटे चाइल्ड रो रन करा जाय आ संगे केते परसेंट मैच करथे कारण जेतबेले रिकॉम्बिनेशन हिताय सेतबेले तमरो सबुतिको जीन न किछि सब कोन जोगुडा का हमर सब रहउ नै कि नॉन कोडिंग एरिया ऑफ द जीनोम सेगुडा को जो मिनी सैटेलाइट मैक्रो सैटेलाइट लॉन्ग टैंडम रिपीट्स सेगुडा सब आसीताय फादर्स रु चाइल्ड फादर मदर्स रु चाइल्ड तो आ संगे सिमिलरिटी एई टाइप रो बैंड दो तो एई टाइप रो जदी सिमिलरिटी बैंड दउची ताले हमें जानिबु जे आर फादर मदर कर ये हला चाइल्ड बेली जदी हमें जानिबु जे रेपिस्ट केसेस किए से जगह रे तो हमें कोन करू ना से जगह रे जो डीएनए थाए से डीएनए को हमें कोन करू ना एम्प्लीफाई करिवु कॉपी नंबर बढ़ाइबु कारण जेहेतु लो अमाउंट रो हमें पाइथबु गोटे दुईटा हेयर पाइथबु कि अल्प सीमेन पाइथबु कि ब्लड सैंपल पाइथबु से ब्लड सैंपल को तुमको पीसीआर करिकि ताक पॉलीमरेज चेन रिएक्शन ने 2 टू द पावर n ने तमे कॉपी नंबर करि परिबो तमे जेते साइकिल रन करिवो सपोज 4 साइकिल रन करला तो कॉपी नंबर विल बी 16 तमे जदी 32 साइकिल रन करला मैक्सिमम हमें 40 साइकिल पर्यंत जाई परिया तमे भाबो डीएनए रो केते कॉपी नंबर इंक्रीज करिवो सेही कॉपी नंबर को कोन करिवो ना हमें एठी जो जेल ले रन करिवो प्लस जो पेशेंट सब की कल्प्रिट माने जो माने धरा आइथिबे तांकर ब्लड सैंपल आनी की ड्रॉन करिवो जदी पूरा मैच करछि परफेक्टली मैच और 80 टू 90% मैच करछि ताले हमें एटा कंफर्म हे परिबो कि सेटा से कल्प्रिट बोली तो सेटा हमें इलेक्ट्रोफोरेसिस रे जानि पारो फिंगरप्रिंटिंग करी
यस सर
सर जो हमर जो रॉयल सर जो डिफरेंट टाइप सर ब्लॉटिंग जेमति कि वेस्टर्न ब्लॉटिंग नॉर्दर्न ब्लॉटिंग और सदर्न ब्लॉटिंग सर ए तीनटा भीतर सर डिफरेंस ता कोन होछि
जेमति कि हमें एबे वेस्टर्न ब्लॉटिंग रे हमें प्रोटीन को टारगेट करूछु माने ओनली फॉर प्रोटीन एक्सप्रेशन सेही प्रोटीन टा हमर जेमति कि हमें सपोज म एबे तुमको एग्जांपल कहूची हमें जतबेले एचआईवी केसेस रे एचआईवी केसेस रे आग एलाइसा करा जाय एंजाइम लिंक्ड इम्यूनो सर्वेंट आसे सेतरे आग डिटेक्ट हला पर एचआईवी रो प्रोटीन्स तापर कन्फर्मेशन पे जाए सबु बेले वेस्टर्न ब्लॉटिंग दिस इज कॉल्ड क्वांटिटेटिव टेक्निक्स तमे क्वांटिटेटिवली डिटेक्ट करूचो सेही प्रोटीन नो एक्सप्रेशन जदी तुमको भाइरस रो अफेक्ट करिची तम पाखरे सेही प्रोटीन टा अधिक आसीबो सेही प्रोटीन टा जदी अधिक आसीबो से प्रोटीन टा अधिक आसी बोली केम जानिबु ना हमें सेही प्रोटीन को एठी वेल ले लोड कर जोठी से प्रोटीन टा अधिक आसीबो से प्रोटीन अगेंस्ट रे हमें कोन करिवु ना प्राइमरी एंटीबॉडी दउ कारण प्राइमरी एंटीबॉडी बाइंड करिवो सेही प्रोटीन को पूरा स्पेसिफिकली कारण हमें जानि जे एंटीजन एंटीबॉडी आर स्ट्रांगली दे आर रिएक्टिंग कारण इम्यून सेल से माने पूरा डिटेक्ट करिवे जोठी के एंटीजन एंटीबॉडी सेही बाइंड करिवे सपोज एटा हला हमर ए रेड कलर ता हला एचआईवी प्रोटीन तो ए एचआईवी प्रोटीन एक्स प्रोटीन अगेंस्ट रे हमें कोन करिवु ना प्राइमरी एंटीबॉडी तयारी करतु सेही प्राइमरी एंटीबॉडी ता आदरे पकेले ये कोन करिवो ना ए एक्स प्रोटीन रेड कलर प्रोटीन को बाइंड करिवो बट आमे जानि परिबुनी आमे केमि जानिबे एक्सप्रेशन अधिक आइछि कि कोन जेतेबेले आमे एठी दबु तो सेतेबेले कोन करिवु ना आको आमे प्राइमरी एंटीबॉडी को कोनी कोन करथबु ना सेकेंडरी एंटीबॉडी प्राइमरी एंटीबॉडी केमि दबु सेकेंडरी एंटीबॉडी केमि तयारी करू सपोज मु तुमको एग्जांपल कहूची जेमते कि तमे जदी ह्यूमन न गोटे प्रोटीन नै कि माउस रे ताको इंजेक्ट करिवो तले माउस पे सी कोन हे जिवो ना गोटे एंटीजन हे जिवो तो से माउस कोन करिवो ना से ता अगेंस्ट रे प्राइमरी एंटीबॉडी तयारी करिवो सेही जो प्राइमरी एंटीबॉडी तयारी करिवो सेही प्राइमरी एंटीबॉडी की पुनी तमे माउस रु बाहर करिकि जदी नै कि रैबिट रे इंजेक्ट करिवो तले रैबिट कोन करिवो ना सेही प्राइमरी अगेंस्ट रे से कोन करिवो ना सेकेंडरी एंटीबॉडी बनेइबो कारण ता पे सी एंटीजन तो एमिति हेले प्राइमरी और सेकेंडरी एंटीबॉडी तयारी हुए जो प्राइमरी एंटीबॉडी कोन करि परियो ना ए एक्स एचआईवी प्रोटीन ने बाइंड करिवो and those secondary antibody with a fluorescence tag it will bind to the primary antibody fc regions this is specific and it will give a fluorescent signal the higher the fluorescent signal it will give the expression pattern and this is low expressed or medium expressed or high expressed then you will find the patient is hiv or not this is called western blotting तो आमे देन आमे पढिथिले कोन ना नॉर्दर्न ब्लॉटिंग रे आमे स्पेसिफिक जीन को एनालाइज करू स्पेसिफिक जीन्स मींस जति एक्स जीन्स गोटे प्रोटीन दउची हमर सपोज मसल प्रोटीन टाइटिन दउची ए जो एक्स जीन जो मसल प्रोटीन टाइटिन दउची मे बी तार आमे काहे किया को ए जीन को स्टडी करूछु ना हमर मसल तयारी होची कि नै बॉडी रे जानिया पे जदी तमरा ए जो एक्स जीन ता म्यूटेटेड हे जिवो ताले ये कोन करिवो ना गोटे याउ दबनी तार एमआरएनए कारण डीएनए रु एमआरएनए हबो एमआरएनए रु प्रोटीन हेला परे जाई टाइटिन प्रोटीन बनिला परे जाई मसल बिल्ट अप हो जदी बट तमरा स्पेसिफिक जीन कोन हेची ना तमरा म्यूटेटेड हे जाई ताले ये कोन करिवो ना उ एमआरएनए दबनी और ट्रंकेटेड एमआरएनए दबो ट्रंकेटेड इन एमआरएनए मींस नॉन फंक्शनल एमआरएनए जदी नॉन फंक्शनल एमआरएनए देला ताले ये कोन करिवो ना प्रोटीन सिंथेसाइज करिवोनी जदी प्रोटीन सिंथेसाइज न करिवो ताले तमरा आ कोन करिवो ना तमरा आ तमे जानि परिवोनी तमरो मसल बिल्ट अप हो आ होपनी कारण जदी प्रोटीन न हो मसल बिल्ट अप होनी तो सेति पे पर्टिकुलर जीन न फंक्शन जानिया पे आमे कोन करू ना नॉर्दर्न ब्लॉट करिकि तार आरएनए को एक्सट्रैक्ट करू जो आरएनए को आमे कोन करू ना इलेक्ट्रोफोरेसिस करिकि ता अगेंस्ट रे सेही टाइटिन प्रोटीन अगेंस्ट रे लेवल प्रोब दबु आ देखिबु जे सेही एमआरएनए रो एक्सप्रेशन कोन है से बढ़ीची ना कमीची जदी एमआरएनए रो एक्सप्रेशन कमूची ता माने से जीन ने किछि ओडे डिलीटेरियस म्यूटेशन है जदी आमे जानिबा आमे देन कोन करिवु ना जदी हेइथिबो ताले आमे क्रिस्पर cas9 ने एडिट करि परिबु एबे तो एडिटिंग टेक्निक आसिलानी तमे किछि मे भी इंसर्ट करिकि से जीन को करेक्ट करि परिबो तो नले तमे कोन होबो ना एटा डिजीज केस को प्रमोट करिबो नले तमे किछि ड्रग खाइकि से जो जीन को ओवर एक्सप्रेस करइबो कारण तमर रेस्पेक्टिव एमआरएनए एक्सप्रेशन पे जदी एमआरएनए न होबो ताले तमर प्रोटीन सिंथेसाइज न होबो नेक्स्ट कोन समटाइम्स कोन हुए ना एमआरएनए जीन ठीक थिबो बट एमआरएनए रे प्रॉब्लम हे जाउथिबो एमआरएनए कोन होइथिबो ना डिग्रेड हे जाउथिबो तो सेही जगह रे भी आल्सो आमे एटा जीन एनालिसिस करिकि नॉर्दर्न ब्लॉटिंग रे करि परिबो सेम हैला सदर्न ब्लॉटिंग रे आमे कोन करू ना डीएनए को एनालिसिस करू आमे सेठी पर्टिकुलर डीएनए को नै कि प्रोबिंग करिकी सेठी डीएनए को एनालिसिस करिवो जदि हमर डीएनए देखिबो जदि ठीक अछि देन नॉर्दर्न ब्लॉटिंग करिकी हमें तार आरएनए को एनालिसिस करिवो जदि नॉर्दर्न ब्लॉटिंग रे आरएनए ठीक अछि ताले हमें वेस्टर्न ब्लॉटिंग रे देखिबो ताले प्रोटीन न कोन हिची प्रोटीन ठीक से एक्सप्रेस होउची कि नै तो एटा हला टोटल प्रोसीड्यूर southern then northern then western blotting. These are blotting techniques.
Good evening, sir.
Good evening.
Sir, मोर क्वेश्चन थिला कि सर आमे जेमति एसटीएस पेज यूज करछु प्रोटीन सेपरेशन ପାଇଁ ठीक अछि जोटा कि मास और चार्ज ऊपर डिपेंड करूची तो सर मु एसटीएस पेज कोन डीएनए सेपरेशन ପାଇଁ यूज कर परिबी
ना एसडीएस पेज हमें डीएनए कारण डीएनए पाखरे ऑलरेडी नेगेटिव चार्ज अछि ना
आगे
हमें कहीं ताकू चार्ज दबा प्रोटीन पाखरे चार्ज नहीं जदि प्रोटीन पाखरे चार्ज नहीं ताले प्रोटीन विल मूव स्लोयर तो आमे जदि ताकू गोटे किछि चार्ज दउचे गोटे इलेक्ट्रिकल फील्ड अप्लाई करले कारण हमर आमे जानि जे नेगेटिव रु पॉजिटिव और पॉजिटिव नेगेटिव मूवमेंट हो तो जदि विल गिव ए चार्ज टू द प्रोटीन इट विल मूव फास्टर देन नॉर्मल प्रोटीन तो सेति पे हमें दबा बट इन केस ऑफ डीएनए रे जेतु फॉस्फेट अछि फॉस्फेट बैकबोन इट विल गिव टू नेगेटिव चार्ज टू द डीएनए सो इट विल मूव अकॉर्डिंगली तो सेति पे के हमकु दबा को पड़िबने वेस्टर्न ब्लॉटिंग केवल प्रोटीन केसेस रे हमें चार्ज प्रोवाइड करू प्रोटीन को कारण प्रोटीन में मिक्स चार्ज थिबो पॉजिटिव चार्ज अमीनो एसिड इज देयर नेगेटिव चार्ज अमीनो एसिड इज देयर हाइड्रोफोबिक अमीनो एसिड इज आल्सो देयर तो सेति पे के विल गिव चार्ज टू प्रोटीन फॉर मूवमेंट ऑफ दिस प्रोटीन्स
ताहले सर मु नेटिव पेज यूज कर परिबी
native page you can use native page only for the enzymes or your antibody antigen reaction in that cases you can use native page not for HTC and in native page it will take 12 to 20 hours it will take क्यों ना movement is very slower because we cannot give any charge to the protein and it will move slower and during the movement of slower movement it will give a friction or it will give generate heats and this heat can degrade that proteins we can only run that native page only in 4 degree or in refrigerated condition but here you can give the charge to protein in that cases it will move faster now first of all तमर टारगेट कोन मु एंजाइम्स को करिवि ना मु प्रोटीन्स रो एक्सप्रेशन देखबि इफ यू वांट टू check the expression of protein then you will go for this SDS page if you go know the enzyme substrate reaction or antibody antigen reaction then you will go for the native page native means in the native form of the proteins here we cannot use the this SDS sodium rhodocell sulfate
आउ सर ये जो पिक्चर अछि हमर बैंड टा वेरी करूची एसडीएस पेज रे किछिटा थिक बैंड अछि किछिटा
हाँ
थिन बैंड अछि सेटर
हाँ
वेरिएशन सर केवल माने नंबर्स ऑफ जो मॉलिक्यूलर वेट जो सेम
expression
मॉलिक्यूल
yes yes तम केते अमाउंट ऑफ प्रोटीन से जगह रे अछि सेही मॉलिक्यूलर वेट रे केते नंबर्स ऑफ प्रोटीन्स अछि तो तार एक्सप्रेशन ऊपर डिपेंड कर
आज्ञा सर थैंक यू
अगर डाउट अछि कोन आउ कोन डाउट अछि आउ किछि डाउट अछि की पचार मो पीपीटी शेयर कर सला परे तमे डाटाबेस को जाई परिबो आरो लिंक भी अछि
से डाटाबेस
पीपीटी
गुडाको माने टाइम टू टाइम अपडेट होउची ना माने केवल स्टैटिक सेमि रहउची
नहीं 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 टाइम टू टाइम अपडेटेड तमे जदी को भी डाटाबेस देखिबो जेते रिसर्च होउची सब ऑल रिसर्च आर मर्ज्ड विथ दिस एनसीबीआई databases NCBI and Uniprot databases. These are the large databases. These two are NCBI and Uniprot. ସବୁ data ତା ଦେହରେ integrate ହଉଛି ଆଉ every time every second ରେ updated ହଉଛି
ସାର୍
କାରଣ
ଏଗୁଡାକ ସବୁ
world wide ଯାହା research ହବ ସବୁ ଏଇ database ରେ update ହବା mandatory or these are linked to every research journal, so all the data are updated.
ସାର୍ ଏଗୁଡାକ ସବୁ ଫରେନ୍ ଡାଟାବେସ୍ ନା ଇଣ୍ଡିଆନ୍?
ଏଗୁଡାକ ସବୁ foreign databases, ସବୁତିକ ଇଣ୍ଡିଆନ ମାନେ ହିଁ ତିଆରି କରୁଛନ୍ତି।
ୟେସ୍ ସାର୍।
ସବୁଠାରୁ maximum ୟୁଜ ହୁଏ NCBI databases ଯୋଉଟା କି ନ୍ୟାସନାଲ ଇନଷ୍ଟିଚ୍ୟୁଟ ଅଫ ହେଲ୍ଥ, ଆକୁ ଯୋଉଟା କି ସମସ୍ତେ ଫଲୋ କରନ୍ତି, ଯୋଉଟା କି ମୁଁ ତମକୁ ଦେଖେଇଥିଲି NCBI ଯଦି ଦେଖିଥିବ NCBI ଏଇଟା ये देखो नेशनल लाइब्रेरी ऑफ मेडिसिन, नेशनल सेंटर फॉर बायोटेक्नोलॉजी इंफॉर्मेशन, एठी तुमको सबु इंफॉर्मेशन मिलिबो, सबु जहा इंफॉर्मेशन खोजिबो, तमे जीनोम खोजो, तमे प्रोटियोम खोजो, सबु एसएनपी एसआरए सबु स्ट्रक्चर टैक्सोनॉमी टूल किट सबु जहा चाहिबो सबु एठी अपडेटेड थिबो। जदि तमे ऑल को जीवो, सपोज मुबे तुमको गोटे यदि एग्जांपल देबी, सपोज मो ना मारिबी एठी देख जदि मु किछि भी मो ना मारुची ताले मोर जहा भी एठी पब्लिश हेइथिबो, एठी जहा भी डाटा मोर पब्लिश हेइथिबो, तो एगुडा को सबु मोर अपडेटेड थिबो ए डाटाबेसेस सबु अपडेटेड अछि। तो सेति पई कि तमे किछि भी कोठी भी गोटे भी तमे डाटा मिस करि परिबोनी, एगुडा सबु लिंक्ड एंड अपडेटेड, ऑल आर अपडेटेड। तो तमे कोटा लास्ट 5 वर्ष 1 ईयर 2024 सब सबु एठी सब थिबो 2023 2024 जहा डाटा खोजिबो सबु अपडेटेड थिबो एठी सीक्वेंस। तमे जीन सीक्वेंस भी जदी खोजिबो एठी जीन सीक्वेंस भी मिलिबो। तो ए डाटा गुडा को एवरी सेकेंड रे अपडेटेड होंती, जेते डाटा पब्लिश हुए गुडा सबु लिंक थान, all are linked. This PDB also linked to this NCBI databases, this Uniprot also linked to this databases. तो एगुडा को सबु थिबो तमे पाइबो कंपलसरी। तो all databases are updated. तो कार डाउट
ସାର୍ ସେ ଯୋଉ ଡାଟାବେସ୍ ରେ ଆପଣଙ୍କ ଇନଫରମେସନ ଆପଣ କଣ ତାଙ୍କ ସହିତ କଣ୍ଟ୍ରାକ୍ଟ କଲେ ନା କେମିତି ସେଇଟା ପବ୍ଲିଶ ହେଲା?
ना 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 आमे सपोज किछि भी गोटे स्टडी करलु आउ तार न्यू रिजल्ट फाइंड आउट करलु, तो सेही न्यू रिजल्ट को हमें कोन करू गोटे गोटे जर्नल ले इंटरनेशनल जर्नल ले पब्लिश हुए, सेही इंटरनेशनल जर्नल उडा को सब ए डाटाबेस सहित लिंक्ड। तमे जहा नुवा डाटा पब्लिश करूचो दे आर लिंक्ड टू दिस एनसीबीआई और यूनिप्रोड डाटाबेसेस, तो से ऑटो अपडेट हिची से
Yes,
अनुसार
sir.
से कोड हिची,
Yes, sir.
तमे संगे संगे से डाटाटा सीटी पाई परियो। तमे जदि चाहिबो को प्रोटीन नो 3 डायमेंशनल स्ट्रक्चर जानिया पई तले प्रोटीन डाटा बैंक रु तमे आराम रे जानि परियो से प्रोटीन नो 3 डायमेंशनल स्ट्रक्चर तादर केते अल्फा हेलिक्स अछि केते बीटा शीट्स अछि छोडा को जानि परियो। तमे
Sir,
पीडीबी
ଏଇଥିରେ
डाटा बैंक
ଯୋଉ କଲର କୋଡିଂ ଯୋଉ ହେଇଥିଲା ସେ ବହୁତ ଗୁଡେ ସେଟା ଲେବଲିଂ ତ ହେଇନି, ଏଟା କଣ କୋଉଟା ଟିକେ ସେ
एई जो कलर कोडिंग
यस सर।
हाँ एगुडा का देखिना बुक रे एगुडा सब बीटा शीट्स गुडा को एमति लिखिथिबो आरो फॉर्म रे थिबो, कारण गोटे ऊपर गोटे सीट फॉर्मेशन करथिबो जेंति एस्बेस्टस को गोटे ऊपर गोटे रखि दबो ना सीट फॉर्म रे रहइबो नले पेपर रे कारण जेतोले हमर अमाइनो एसिड थाए अमाइनो एसिड बहुत वास्ट थाए छोड़ा जो सीट फॉर्म रे गोटे ऊपर गोटे थाक होई की रहिबे तो सेति में एटा सीट पे आरो मार्क थाए आ ए जो कॉइल फॉर्म रे जो गुडा थाए सेडा सब हेलिकल फॉर्म तो से गुडा थाए हेलिक्स आ जदी गोटे हेलिक्स आ गोटे सीट को जोड़ी हेइथाए गोटे मोटिव्स द्वारा और टर्न द्वारा एटा कोंदी हेलिकल टर्न हेलिक्स रु सीट्स दीटा का अटैच हेइथि हेलिक्स आ सीट दीटा का अटैच हेइथि तब कोंदी हेलिकल टर्न
यस सर
तो एगुडा करे तमे फंक्शनल पाथवे एगुडा को स्टडी करि परिबो पाथवे कोठी पोजीशन बायोलॉजिकल प्रोसेस रे कोन तार अछि मॉलिक्यूलर फंक्शन सेलुलर कंपोनेंट को पाथवे रे मान इन्वॉल्व ए जे स्ट्रिंग डाटाबेसेस अछि जानि परिबो पीडीबी रे ओनली प्रोटीन कथा जानि परिबो तापर जदी तमे ऑल डाटा जहा भी डाटा जानिए को चाहछो तले तमे एनसीबीआई नेशनल लाइब्रेरी फॉर मेडिसिन यूज करिकी तमे सर्च करि परिबो जहा ना सर्च करिवो ता डाटा मिलि जो भी साइंटिस्ट जो भी रिसर्चर हुंतु तमे ताको जानि परिबो इजीली तमे इठी पाइजिबो नेशनल लाइब्रेरी ऑफ साइंस एठी तमे इजीली तार ना डिटेल्स कोठी से काम करिसंती सबु डाटा आदरे थिबो तांकर तमे सेउटू तमे जानि परिबो तम डाटा उडा तापर तमे यूनिप्रोट रे ईच प्रोटीन जेते प्रोटीन डिस्कवर हेची तार सीक्वेंस तार फंक्शन तार पोस्ट ट्रांसलेशनल मॉडिफिकेशन एनी डिजीज और वेरिएंट्स of that protein all data you will find in this uniprot databases तो तुम माने पीपीटी रू भी लिंक अच्छी जाई परिबो नले सेउटू जाईकी डायरेक्ट भी गूगल रे सर्च करिकी तमे करि परिबो कारण एगुडा को सब फ्रीली अवेलेबल दिस ऑल
सर
दिस
गोटे
डाटाबेसेस
सीआरपी गोटे सी रिएक्टिव प्रोटीन गोटे थाए
सीआरपी लेवल
से आज्ञा से
सीआरपी
गुडा
भी
को
तमे एटी भी तमे जदी सीआरपी प्रोटीन भी खोजिबो एठी देखो सीआरपी कोन कोन करूछ सी रिएक्टिव प्रोटीन देखो ता ऊपर केते रिसर्च हेतिबो देखो सी रिएक्टिव प्रोटीन रिएक्टिव प्रोटीन तो ये जो सी रिएक्टिव प्रोटीन कार्डियोवास्कुलर डिजीज तापर कोड सब कोरिलेट करिवो सेगुडा सब तमे पाई परिवो सी रिएक्टिव प्रोटीन न यूनिप्रोट रु जदी नबा ता नेम टा सी रिएक्टिव प्रोटीन असला सी रिएक्टिव प्रोटीन तमे सेठी क्लिक करिकी ह्यूमन को जदी जीवो एटा तार आईडी तमे एठी को फंक्शन ए कोन करिवो एग्लूटिनेशन करिवो स्वेलिंग करिवो फागोसाइटिक कॉम्प्लीमेंट फिक्सेशन थ्रू इट्स कैल्शियम डिपेंडेंट सिग्नलिंग ऑल डिटेल्स तमे से सी रिएक्टिव प्रोटीन्स रो सब पाई जीवो कौन करिवो acute phase response, inflammatory response, innate complement components कारण एगुडा C reactive protein बहुत related to cardiovascular diseases तो सेति कि तमे जो C reactive protein न भी तमे सब फंक्शन जानि परिबो आ ए जो CRP अछि तार जदि देखिबो एठी CRP पुट करियो एठी आसिला देखा C reactive protein कोठी होउची कोन कोन रिसर्च चलची ए जो CRP ऊपर तमे ओडा सब C reactive protein ऊपर जानि पारि CRP
এটা স্যার
कोठी केते भैरी
ইন্টারন্যাশনাল বলবো এটা
ये
ইন্টারন্যাশনাল
सब इंटरनेशनल ये टोटल इंडिया as well as इंटरनेशनल जहाँ भी रिसर्च चलती वो सब सी रिएक्टिव प्रोटीन ऊपर ए डाटाबेस रे तुम पाई जिवो जहाँ भी खोजो तुम किछि भी प्रोटीन जहाँ भी स्टडी करो सपोज थायराइड ऊपर किए जानी हो चाहूँ थायराइड थायराइड ग्लैंड की थायराइड कैंसर रु जे जानी हो चाहिबो तमे एटी आसीबो न्यू बायोमार्कर एबे डिस्कवरी होउची एटा केबे कर 23 रो तमे जदि अपडेट जानी हो चाहिबो अपडेट देखो 24 रो अछि पेपर जुलाई रो ए रिसेंट पेपर ये सबु मिलिबो तुमको ए डाटाबेस रे ऑल डाटा वर्ल्ड वाइड जहाँ भी रिसर्च जोठी भी रिसर्च चालू तो ए डाटा सहित समस्ते इंटीग्रेटेड ऑटो इंटीग्रेटेड तो किछि ये करि परिबोनी तो अदे तमे जा जो भी कैंसर जानियो तमे थायराइड जानो कोलन कैंसर जानो ए डाटाबेस पे सब डाटा पाइबो लेटेस्ट रिसर्च कोन चलची कोन ड्रग ट्रायल चलची सब जानियो एटी कोरियन पॉपुलेशन ने कोन क्लिनिकल प्रैक्टिस गाइडलाइंस होची डॉक्टर माने भी आकु फॉलो करंती डिफरेंट सोसाइटी एक केस पर Cas9 रे जीन एडिटिंग होची कोलन कैंसर ट्रीटमेंट पे तो कोन कोन सब लेटेस्ट रिसर्च चलची all data you can find in this NIH library sites जो कैंसर जहाँ भी जो भी डिजीज ये जानिए को चाहिए all the disease and information are available in this National Library of Medicine that is PubMed जोटा कि हमें describe करी जो एट NCBI तो क्लिक कर दबो NCBI गूगल ले तो जहाँ डाटा जानियार अच्छी रिसर्च की मेडिसिन कौन तापे अच्छी तमे से डिजीज पे तमे क्लिक करो नेशनल सेंटर फॉर बायोटेक्नोलॉजी इंफॉर्मेशन देन यू कैन क्लिक दिस एटी अछि ऑल डाटाबेसेस कौन कौन जानि हो जाउछो तमे को ड्रग रो ट्रीटमेंट ऑप्शन जहाँ भी ड्रग पे कौन ट्रीटमेंट अच्छी सब आसी सपोज मु एटी टाइप करी मलेरिया प्लस पैरासिटामोल कौन यूज होची देखो मलेरिया और पैरासिटामोल ऊपर कौन चली कोटा फेज 2 ट्रायल ले चली को ड्रग टा सब आसी जिवो एठी तुम पेन मेडिकेशन ने केमती करिवो पैरासिटामोल इन फार्माकोकाइनेटिक्स प्रॉपर्टीज ऑफ इंट्रामस्कुलर वर्सेस ओरल सिरप माने ये इंट्रामस्कुलर और वर्सेस ओरल सिरप पैरासिटामोल इन प्लाज्मोडियम फाल्सिपारम मलेरिया से केसेस रे केमती काम चलची सब डाटा तमे एठु पाई परिबो को मेडिसिन भी चलची लेटेस्ट को मेडिसिन न डिस्कवर भी होची सब एठी ही सब मिलेबो तुमको एटी अछि क्लिनिकल ट्रायल्स एटी क्लिनिकल ट्रायल्स को न्यू ड्रग 250 new drugs are in clinical trials for this malaria एटी अछि क्लिनिकल ट्रायल्स तमे उड जानि परिबो एटी जीन्स केते जीन वेरिएशन होची केते जीन डाटा सेट सबमिट हेची एगुडा भी तमे एठु पाई परिबो आमे जो ओएमआईएम एगुडा सब डाटाबेस देखतले एट सब अछि ठीक अछि आज
Yes, sir.
क्लास क्लोज
Yes, sir.
करिबा
Yes, sir.
ମୁଁ पीपीटी भी शेयर कर देबी
Yes, sir.
कुछ करिबा तो ओके
Yes, sir.
थैंक यू
सर काली हम लास्ट क्लास
थैंक
रहइबो
यू सर।
हाँ काले लास्ट क्लास रहियो
सर आरो କିଛି क्लास नहीं
सेटा तो हमको इन्फॉर्मेशन नहीं तमे सार संग जे कोऑर्डिनेटर थिबे तम सेंटर रो तांग संग कांटेक्ट करियो सी जदि हमको इन्फॉर्म करिवे कोन करिया पे हम कहियो
सर आपण एबे कोठी माने आपनकर ये माने पोस्टिंग
मोर रिसर्च चली रेवेन्सा यूनिवर्सिटी एंड इंस्टीट्यूट ऑफ लाइफ साइंस भुवनेश्वर
आज्ञा आज्ञा
मोर बेसिकली कैंसर ऊपर रिसर्च ब्रेस्ट कैंसर
यस सर
क्लोज करिया से सर
यस सर थैंक यू सर
ओके सर
थैंक यू सर।
थैंक यू सर